Hi guys, my name is Christoph Harmel. I'm a PhD student from Switzerland doing computational biology, and right now I'm in the late game of my PhD, so like 4th year. And I'm going to tell you something about multimodal spatiotemporal phenotyping of human retinal organoid development, so like a lot of punchlines in here, like buzzwords. And I'm from the group of Greg Kemp. We are also like a developmental biology group, doing also a lot of computational biology, and we just, like 2 years ago, we moved to the Institute of Human Biology, which is a Roche founded institute, which is like focusing on developing organoid systems and like bringing organoids to the point where they can actually be used in pharma for like safety testing, model improvements, drug screens, and stuff like this. And yeah, today I will mainly focus on this paper we published like almost a year ago by now. And if I have time later on, I will also like show what I've been doing since then, like on the computational sides, like the tools I developed for this stuff here, how I like improved it and applied it to some other data sets and stuff. But let's see how we go time-wise. So maybe just for starters, like a tiny introduction to organoids. So what are organoids? So in principle, like organoids allow you like to either from like tissue-derived cells or there's like inducible pluripotent stem cells, take them, seed them in culture in a 3D environment, like most often like using matricial, for example, and applying like the right morphogen compositions and chemical stuff, and adding some time. Then in the end, like you have like a self-emergent system, which is like really recapitulating phenotypes of the actual real tissue you derive it from, or like you use like certain morphogens to push it into a certain fate of a tissue. And for example, here this would just like be a cartoon of the intestinal organoids where you have like the crypts and the gut. And I mean, this we basically did for the eye, so it's already here the disclaimer. And in addition to this, in addition to this, like what we are aiming to do is like really from the multi-omics perspective, look at such developing systems. So what we are doing mainly in our lab is like we do a lot of single-cell sequencing on RNA, also ATAC and multi-ohm data, and also a lot of multiplex imaging. So mainly immunohistochemistry data sets where we can like reach plexicity of like 60 antibodies or even up to 100 antibodies on FFPs. Right? And my main task here was then like to bring like such data sets where you like have a temporal component and this like multimodal component, but from different experiments together in like one representation. So like working on like digital multi-omic representations of organoids. That's the whole aim here, like reaching this like endpoint of multimodal spatiotemporal integration. So in the case of the retina, what we are doing is we take iPSCs and then grow them into retinal organoids and keep them in culture over a time course from over 39 weeks. So it's a pretty long time. So congrats to like the wet lab people there. So like I have to maybe like quick notify, like I didn't do like any wet lab myself. So like there were like 2 postdocs being involved in this. Like one guy just like optimizing the whole imaging and sample handling, and that was Philip Bali, and Giovanna Barcati, who did like all the sequencing, which I will be presenting here. And another postdoc, Jisong He, who also helped with the computational work and was more focused on the single cell data, and I was more focused on the imaging data. But here what you can see, like our approach is like we grow these organoids, and we also have like primary tissue sections, and then we place them on a 96-well structure, and then we do this like reiterative round of staining. So like we always put 3 antibodies on top, do also Darby staining, image the thing, elode, repeat, rinse and repeat over like a month or so. And then we come up with this like very beautiful data set where we really go from like kind of really like millimeter scale to nanometer scale with 40x confocal imaging, with like oil and everything. It's like really nice imaging quality. And already like just from the protein stains, you can see that like the structure you would normally see in an eye, like having like cones and rods on the outside, having the Muller glia, the retinal ganglion cells, amacrine cells, bipolar cells. You all have them like nicely spatially organized in the same way as you would have them in the adult retina, for example. So like this is the primary tissue here, and that's a slice from our one of our organoids. So in total, like in this data set, I guess we had like 45 stains and up to like 30 or so organoid slices. So rather big data set actually. And now just like an overview here, like it's a lot of data. So like you cannot really grasp like all the images and just like making sense of just creating overlays. It's overwhelming, too many permutations in there. So what we came up with is that we perform a multiplex tissue units analysis. And this is kind of like informed or like inspired by what we do in single-cell RNA sequencing, where like you would have like one cell, in this case it's just a pixel, and then like on this like n-dimensional space of features, in this case the protein stains, in single RNA seq it would be like the transcriptome, you perform clustering, unsupervised clustering. And that's what you see here. So like each color here would correspond to like one pixel cluster. And then if we then like put these colored pixels regarding for the clusters back into the image, we come up with such representations where you actually get like biological meaningful clustering of the system. So you would have an MTU which is marking Muller glia nuclei, one MTU which is marking amacrine cells. You see like collagen deposits in the system. You see cones and rods and stuff like this. And the cool thing here is that, I mean, we have like the complete time course of data, right? So like we can actually see like this laminated structure of the retina forming up over the time course. So like here we start from 6 weeks, it's like a little bit disorganized. And then around like 18 weeks, you already start getting a sense of the lamination here over 24 weeks to the final endpoint in our data set, which was like 39 weeks. And this data point is then like super close to the actual like primary system. But I mean, after all, this is like just like, it's not quantitatively looking at the data, it's just like getting it into another representation and like making sense of it from your like biological knowledge you have, right? But what we actually wanted to do is like really put numbers onto like the developmental stage of the system from week 6 to week 39. And how we actually did this is like we thought like, okay, we have this very stereotypic spatial organization on the outer layer of the organoids, which is corresponding to the primary tissue. That's what we see. So, and what we're doing now is like, or that's mainly my part here, that I developed this algorithm called Laminator, because we laminated stuff. And we, on the outline of the organoids, we basically just place rectangles oriented to the center of the organoid. And then we read, like along this axis, like the inner outer axis of the laminar windows, we read out the intensity profiles from all the protein stains we gathered. And from this information, then we are able to reconstruct pseudotime trajectories, like falling back to like diffusion map algorithms, for example. And this allows us then to like reconstruct a trajectory which reflects the development of the system. And then you can like just show it by making collages of these MPU images for like different time points, for example, where you can see like it's like nicely ordered and the structures make sense, or like having the complete overview by like creating heat maps over this trajectory and then looking at either MTUs or just like nuclei stains, for example, like here the Höchst stain for the nuclei. That's like in the beginning it's very disorganized, then over time it's like really forming this double layer, like the inner and outer plexiform layers of the retina. And then like if you put numbers on this, then you can say like, okay, we can come up with a maturation score where we show that the later time points of the organoids are actually like becoming closer in their composition of the laminar structure to the primary tissue, which are like here, the red dots.
So PT on an x-axis is time, or what is
it? Yeah, pseudo-time. Yeah, so like just like ordering, like finding a path through the diffusion space and then ordering the points in there. It's mainly just like, here that's like the embedding for the diffusion maps. And then you see like the pseudo-time which you calculate in this labeled on top. This is here now like just like zooming into the algorithm. I mean, mainly what you get is like from all these laminar windows, you come up with like for each protein stain, like have like you can say like a time signal, but I mean it's in space, but like just like that. And then I mean I do some like processing, like I downsample, I smooth the signals, and then as you can imagine, like in the if you want to do this like globally in the complete data set, it's becoming big because in the end what you want to come up with is like one dense distance matrix. And this is computationally intensive, so what I found like that like comparing these signals in n-dimensional space, it's not very effective if you do it with cross-correlation, but rather just like first transforming it into Fourier space, and then in Fourier space do Euclidean distances. And then use these distance matrices to then run the diffusion map algorithm, for example. And then I mean in this space you can also do like unsupervised clustering. And I mean this already helps you just like because organoids can be messy, they can be like the variability is high, so we can already use this algorithm to like just like assess like which regions of the organoid would actually be interesting for us to compare to the primary tissue.
So computational isn't basically cross-correlation Euclidean and Fourier space?
Yeah, it is, but just like computationally it's bigger because like the Fourier transform, it was like in Python, it's implemented in C, and yeah, it was just convenient to use. Yeah, now bringing in like the single cell data. So Shisong did this whole.
So before you go on, I want to make sure I understand what you've done here. So what you're doing is taking a slight cross section of your organoid, right? And then you're looking at the intensity profiles of the different channels, different stains that you have, and then you're quantifying that and seeing how that changes spatially across that section.
Exactly.
And that will tell you whether your organoid is at its final destination.
Exactly. Yeah.
What did you, this diffusion thing, you kind of lost me, what did you say about
the diffusion maps, it's called. It's a dimension reduction method, but it's like in comparison to PCA, it's non-linear. So like, yeah, you're trying to find an embedding due to like diffusion processes which describes your data. It's not linear, but non-linear. Okay, so and next step here would then be like that we have like integrated atlas of retinal organoids, but from the transcriptomic side. So we have like attack data in there, and we have RNA-seq data in there for the complete time course. And I mean there, like just from the cell states, you can also construct trajectories that like from the progenitor cells here, like these develop into Müller glia, ganglion cells, amacrine cells, bipolar cells, like all the cell types I've already shown you. You can also see in the transcriptomic space. Finally, like the UMAP projection of the whole thing is a dog, so I don't know what this says about vision, but so that was now like the data set we wanted to combine with our spatial data set. And and like also like from the attack data here, of course, you can like reconstruct gene regulatory networks. And then also show like which gene programs for which cell type are active, which transcription vectors are important. They are like getting like a very complete picture of the system. And now like coming to the actual integration. So like our idea was like to use RNA as our anchor data set, because I mean for each protein we pretty much know like which RNA transcript would be responsible for its production, and the attack can be mapped to the RNA because like you have like certain gene regions, you know, the gene body corresponds to a certain transcript, so like there's the linkage, like the RNA is our pivot point where we can do like our data integration. And then for the actual integration, we just used like Seurat, like a standard processing pipeline in the single cell world, where you mainly like by overlapping features, like for the protein and the RNA, you do some KNN graphs, and then you, by finding mutual neighbors, you can just like transfer labels over. But I mean, this worked pretty good in our case. So like for each time point of our organoid data, we could label transfer first like the cell type labels, and then also like bringing over the complete transcriptome by just like, if you apply super high resolution clustering in the RNA space, and then you transfer these labels, then you can just like take the average expression for each gene in these high resolution meta clusters and visualize them then back into the tissue. So like then you can see that like for certain key regulators like transcription factors in the retina, they like perfectly nicely in the right layer of the organoid. You can see that like the cell type composition of the whole thing is making sense. So we were pretty happy with that. However, some more stuff. Yeah, sorry. So like now, I mean, I told you that we had all these windows, right? And now we also know like the position of each nuclei in each of these windows. So we can actually now like visualize like our map transcriptome or like attacks peaks or just the cell types in this pseudotemporal space. And then you can see that like certain cell types at the beginning are just like everywhere and narrowing down, or just like are not present anymore, finding the right place in the layers, stuff like this. So like now we have like really integrated data set which we can also like analyze in this trajectory basically. And I mean, up till then here, this was like our first submission, and then the reviewers were like, yeah, but you make an assumption like RNA and protein, and it's not always the same. So for this, we went back then and sent some tissue slides to a company called, or got the name, but they are doing like basically Merfish as a service. So we collected some more organoid slices on fresh frozen tissue, performed multiplex fish, and then my quest was like, okay, now we have to bring like yet another data set into the whole integrated thing. So how we did this is actually we used these like windows again as our like reference coordinate system, where we say, okay, like first of all, I can basically do the same analysis as I did for the 4i data, and then ask, okay, like which profiles I observe in the MERS scope from the transcript stuff is like referring to the predicted RNA profiles I've seen before, and then match these. And I mean, this is then like what you can see here. Like I mean, in the fish data, we had 2 time points, week 13 and week 32. And week 32 was like most closely related to week 24 and a little bit better, less to week 39. And the week 13 was close to the week 12 in the fish data. So that was already pretty promising. Then I mean, for some key regulators, then you can just like look at these profiles along the window and see that in the fish and in the 4i, these are like very much nicely correlated. And then the next step is that you can actually show that the 4i and the fish integrate as good with the actual single-cell RNA-seq data by basically then in these windows doing like nuclear matching. So like you find like the best 2 matching windows, and then you say like, okay, I have nucleus from 4i here, it's in the upper region, okay, so like good partners for this would be also nuclei in the upper region of the fish data. You match these, and then you can just like do correlation. And then you can see that like for transcripts which are high expressed, like the predictions work pretty well. For transcripts which are actually low expressed, it's not working as good. But I mean, this is also like if you just integrate the fish data with the RNA-seq data, there like the predictions are also not good if you have like low transcript counts. Basically, you have a hell lot of noise, and your predictability is just like going down. And in the end, we just asked, okay, like how nicely can each of these data sets, like the fish or the 4i, resolve the actual single-cell RNA-seq data by just like clustering the single-cell RNA-seq in higher and higher resolution, and then letting like the matched nuclei saying, okay, like can they still distinguish like this resolution of clustering in the RNA-seq? And there we've seen that like both the 4i and the fish data perform similarly well. And I mean, this is like strongly dependent on the gene panel you're choosing. I mean, for the fish, and also like the antibody panel you're using for the 4i. And our conclusion was like that mainly we can resolve major cell types, maybe some sub-cell states with both techniques, but you don't reach like the actual resolution you get from single-cell transcriptomics. Yes, what's the time? 5 more minutes? Okay, so it's a huge data set. I actually built an app for that, so it's browsable on the interwebs. Let's see. Oh, no. disconnected. But in principle, like that's our Shiny app where you can browse the complete imaging data, the complete transcriptomics, fish data, everything is in here. It's hosted by the EDH, so feel free to have a look. But this would cost too much time now. Okay. And I mean, we have 5 minutes left, so should I keep going for more computational stuff, or do you just want to ask questions and we discuss it a bit more? I guess
you can keep, yeah, keep going for a bit. It's okay.
Okay, then I keep going. So yet another analysis we did with these systems is just not like using laminar windows, but rather like focusing more on like nuclei, spatial neighborhoods. And I mean, in that case, I thought, okay, like we can actually use the same algorithm, but not for windows, but for circles. And then like say like our inner outer axis from the outside of the circle to the inside of the circle. And in the retinal organoids, actually, there's a deficiency, you might say. So like the ganglion cells, which normally make connections to the brain neurons, at some point they die off. Like after 12 weeks, they're just like vanishing. And we thought like maybe we can like learn something about the neighborhoods, and then I basically did the whole laminator analysis, but with nuclear radial neighborhoods. And we've seen that like there is actually like a heterogeneity for like these RGC cells in the organoids, and actually there are MTUs which are like related to apoptosis, so like high mitochondrial content, wrinkled Darby stains, stuff like this. We could observe, and then we could go back to the single cell data and do like classical DE gene analysis for like these two clusters here, and then we've seen that like in the major populations of the RGCs, like an apoptotic signature was enriched. So I mean, we couldn't solve the riddle why these are dying off, but we can definitely say like by week 12 they die. However, like these assumptions we make here that is like not actually meeting the stuff we observe in the tissue, right? Like if you have a circle and then like the averaging the signal, like it would assume that you have like kind of like a isotropic information in the neighborhood, but I mean, we all know like tissues can be messy, that's most often not the case. So most likely like this method is just like as good as just averaging the signal in the neighborhood. And like these laminar windows in general, they are like highly specific. I mean, in this system you could nicely use it. I mean, you could also maybe use it in like brain slices where you have the different regions or something, but it's not in general applicable to the whole thing or any other biological system. So what I now worked on is working more with like KNN graphs. So like in a tissue, you would have like segmented nuclei. You give like a certain distance threshold and build these graph structures. And then what people nowadays most often just do is like they take the information they get from the nuclei. And in order to bring in like spatial information, they would average over neighborhoods, or they would do something called message passing in graphs. So what this comes down to in the end is just like taking, like in this KNN graphs, you can represent them as an adjacency matrix. So like for each connection, there's a one in the matrix. And then in order to like summarize over neighborhoods, they would just do the dot product of the feature matrix with the adjacency matrix. But I mean, this approach for sure is better than just like clustering the nuclei themselves, but it's missing information in between the cells because it's just like restricted to the thing itself. So what I thought, okay, why not we just like place the laminator windows on these edges and average over these, taking this information and build up like a weighted adjacency matrix where we like actually populate the adjacency matrix in the middle here with the nuclei feature, and then the sides we populate with information we gain from the space in between, and then perform this graph passing, message passing process on these. And this actually worked pretty nicely. So just like an example from the retinal organoids, just after the submission, and like maybe like 3 months old by now, but I like it because it's like really robust. You can like really see that it's like resolving different layers here in the data, and I'm pretty happy. And now I try to like really make this computationally efficient for big data sets, so like billions of cells, and apply it in our latest project. We work on tumoroid data in PDAC. And another thought I had was like, okay, this still is just like observing tiny neighborhoods, like one hop in the what about like long range interactions? How would I capture like morphogen gradients in the tissue, stuff like this? And here I thought like, okay, this data representation might actually be useful because, I mean, you can imagine that like we can build paths in these networks, and each path will be described by the laminated windows, and we just concatenate them. And then we just have like the same data structure and can do the same dimensionality reductions we did before. And this is also just like proof of principle. So what you can then do is like asking like in this KNN graph, okay, I want to find a structure. I want to have restrictions on my structure. So I say like, I want to look for a path which has like setup A linked to setup B, then B is like linked to another setup C, and so on so forth. So like you can like make a query, and then it goes through the whole graph structure and finds like motifs, or in this case just paths, which satisfy this stuff. So that's what I did here. So I like wrote a tiny query, and it found me like these two neighborhoods here. And then if you go ahead and just like do all the distance calculations as we did before for the laminated windows, you can see that like motif versus background, which is orange here, there the distances are significantly higher compared to the background model, where I just like take random paths in the tissue and calculate all the distances, and like the motif versus itself. So that's already like pretty promising that this could be applied. Like if you have already like a biological question in mind and can like cluster your tissue biologically meaningful, then you can basically just look for structures. I mean, and you could also think about this for like graph convolutional networks, learning this de novo, but that's where I am at now. Exactly, and just to the end, acknowledgements to Ray Kemp, Barbara Treutlein, the postdocs involved in this project, and Lucas Parkman's lab in Zurich, who basically taught us like how to do the whole iterative immunohistochemistry stuff. And yeah, Roche, our group, University of Basel, and ETH. Yes, that's it.
Yeah. So this is phenomenally cool. We do a lot of things not necessarily at the genetic level, but at the physiological level.
Yeah.
So we, VMM is one of the, memory potential is one of the big things we care about, but also other physiological indicators. And so I'm wondering, how hard would it be to port this from genes over to sort of something else? Like if you replace genes in space with VMM over time or calcium over time?
Yeah, I guess it's pretty easy. Like as soon as you have like a matrix representation of your data, like images, I mean, it's just a matrix, right? And yeah, and you might have like multi-dimensional measurements in there, then it's applicable for sure.
Great.
The problem with this analysis is this is a fixed data, constant data. The VMM is a time dimensional data.
Yeah. I mean,
that
then
will be a problem.
Yeah, then it's like adding one dimension more, and like the distance calculations and everything would get more complicated, I guess.
Right, but also, as opposed to having 500 genes, if we have like we'd have like kind of five physiological things over time. So in terms of computational stuff, I would guess it's probably quite a bit simpler.
Yeah.
Can you, besides your use, one of the things that we always struggle with is sort of how much is enough biological data, right? So you're always like, I want more samples, more genes.
Yeah.
Can this work in the opposite? Could you do things like drop out individual signals and see like what's the minimum amount that you need to get, right? So if we cut our samples down our number of genes by half, we still get 90% of the predicted value. And that might be good for us for just speed and optimization.
Yeah. I mean, we haven't done this in this project, but in principle, we did some stuff of this. I mean, for the the gene panel design for the fish, for example, there's like I forgot the name of the package, but it's like there are ways to like really like brute force this and come up with a like nice panel where you just don't need to test like everything before, just like test your prediction. It's a good panel. And also, I mean, we did some stuff where we just like dropped out certain proteins and see how the whole thing would be changing for sure. But I mean, in the end, like it really comes to prior biological knowledge, I'd say. Yeah. I mean, in the future, it would be very cool just like having like a system, like phenotype like this, and then you just like go in and say like, I want to do this experiment, like give me the perfect set of parameters. Right? Yeah, exactly.
How big is variability? How many like repeats you need to do so to make sure that that's actually what you see is not just random, so you see that it's like robust.
In which regard?
If you, how many organoids, how many repeats you do for each?
I guess in this study it was like 40 organoids, and I mean we had like 3 replicates per organoid itself, so slices. And I mean that was pretty obvious that it's like highly reproducible. Like I mean, if you wanted, you could just like register the images on top of each other, almost perfectly fit. Because I mean they cut super thin slices, like micro.
But from organoid to another organoid. So
yeah.